Okay, so the next two slides, we're going to be taking a close look at the nephron because this is a topic that the MCAT does like to test. So blood arrives from the body via the afferent arteriole and enters a bundle of capillaries known as the glomerulus. So over here, I underline the A in arrives and the A in afferent. I think that's a pretty helpful mnemonic for you to remember the difference between afferent and efferent arterioles. If you were studying, other systems like the nervous system I did a lecture on, we were talking about afferent and efferent neurons. So it's the same thing. We use the words to connote the same exact thing. Afferent is arriving, efferent is exiting. So in this case, the afferent arteriole connects to a bed of capillaries in the glomerulus. It goes into the glomerulus through those capillaries, and then it comes out the efferent arteriole and travels down the medulla and back up via the vasorecta. So the vasorecta are these sort of capillaries that are following this loop over here, which we'll later talk about, called the loop of Henle. So the pressure in the glomerulus and the glomerular filtration rate, which normal filtration is about 90 to 120 milliliters in humans, is regulated by the constriction and dilation of the afferent and efferent arterioles. So the glomerulus, just like any other capillary bed, is very sensitive and they're very fragile. In order for capillaries to do what they're meant to do, which is exchange nutrients and gas, they have to be super thin. So capillaries, actually, like some of the finest capillaries, blood cells will pass through single file, increase the surface area, so there's gas exchange that's allowed. So they're very fine and they're very fragile. So we have to have very fine-tuned ways to control the blood pressure going in and out of the glomerulus. So one of the ways they do that is control of the afferent and efferent arterioles. So vasoconstriction of the efferent arteriole causes an increase in the glomerular filtration rate. So if you think about that logically, the efferent arteriole is the one that is leaving the glomerulus. So if you constrict that, you're stopping the flow of blood out of the glomerulus, but the flow of the blood in is remaining the same. So that's going to increase the blood volume in the glomerulus, and it's also going to increase the hydrostatic pressure. Likewise, the vasoconstriction of the afferent arteriole causes a decrease in glomerular filtration rate because it's cutting off the blood supply to the glomerulus. So if we cut off the blood supply by constricting the afferent arteriole, and we keep the efferent arteriole open, the glomerulus is going to lose blood volume and it's going to lose blood pressure, and the GFR is going to go down. And the same applies to vice versa. Vasoconstriction of the EA causes increase, vasodilation of the EA causes decrease. Filtrate from the blood is emptied from the glomerulus into Bowman's capsule and heads toward the proximal convoluted tubule. So Bowman's capsule is just that surrounding area of the glomerulus. The filtrate leaks out of the glomerulus and is collected by the Bowman's capsule, which is also known as the glomerular capsule. And it is like funneled in right over here and heads down to the proximal convoluted tubule. There's a lot of information on this slide. Does anybody have any questions about it? Specifically about pressure in the glomerulus and how pressure affects the GFR. Okay. Because we're going to continue a little bit with the nephron on the next slide. So if you guys are okay with this, then we'll move on. So the nephron continued. Filtrate, as I mentioned before, is the fluid that leaks into Bowman's capsule and goes through the entire lumen, this whole loop. It's a fluid consisting mostly of sugars, amino acids, urea, and it enters the proximal convoluted tubule from Bowman's capsule. So picking up where we left off on the last slide, the proximal convoluted tubule is responsible for about 70% of all fluid reabsorption. So sugars and sodium are actively transported back into the surrounding blood vessels, and water passively follows. So it's important to have an understanding of osmosis and how it works. It's not too hard conceptually in terms of the kidney. All you really have to remember is that water pretty much follows where the solute goes. So sugar and sodium gets pulled back up, water is going to follow. The filtrate then enters the descending limb and the loop of Henle. So the descending limb is highly permeable to water. As it descends farther into the medulla, the concentration of the surrounding tissue increases in ion concentration and drives water out of the filtrate. So when we're talking about the descending limb, we're actually talking about this on this diagram over here, this left side. So this top, like we mentioned before, the glomerulus and these top parts of the tubules are in the cortex. So those are isotonic, remember? It's basically the same concentration as blood plasma. So there's not going to be much fluid exchange there. Once you start, well, there's not going to be much fluid exchange passively. You know, we have active transport for ions. So anything that requires ATP can go against its gradient. But in terms of passive transport, as we get farther down this descending limb of the loop of Henle, we get into the salty medulla. So it's high solute, high concentration, and that actually forces water out. Okay, so the thick section of the ascending limb of the loop of Henle actively transports ions back into the medulla to keep its high ion concentration. So the difference in permeability between both limbs to keep the concentration of the medulla constant is known as countercurrent multiplier. And that's a concept that you'll see pop up on the MCAT in a couple of different places, the countercurrent multiplier, countercurrent exchange. The red structure right over here is the glomerulus. That's just that red ball right in the middle. That's where the afferent arteriole connects to the glomerulus, and it filters the blood and creates the filtrate to go to the rest. The red descending structure. If you're talking about this, those are the vasorecta. So the vasorecta are basically blood vessels that travel along the loop of Henle. So this blue, this blue is the deoxygenated blood, and this red is the oxygenated blood. And these purple crossing over is just, you know, the oxygenated blood crossing over, getting rid of the oxygen into the tissues, and then leaving the veins. So like I was saying, the countercurrent multiplier is a concept that's going to pop up pretty often. So in terms of the nephron, we're talking about countercurrent multiplier in that the ascending limb is highly the red is the capillary and the blue is the vein. So it's countercurrent multiplier, I'm sorry, just because water flows out into the medulla, which sort of would lower the solute concentration, but when you loop around and you start going back to the ascending limb, it actually filters the ions back out into the medulla, so it restores the concentration. So it's countercurrent is basically two things that are going in different directions, but it increases the efficiency of the system. So it's just going down natural gradients, and because they're countercurrent, it's efficient. I hope I'm explaining that well enough. If you'd like me to explain it differently or explain it again, then just type in the chat and I will try to explain that again. So the difference, we did that. The filtrate then enters the distal convoluted tubule, where reabsorption of urea, sodium, and water is highly regulated. Secretion of drugs and toxins occur here also. So the distal convoluted tubule, like I said, reabsorption occurs. Reabsorption is when the filtrate, stuff from the filtrate, is being absorbed back up into the tissue. Conversely, secretion is when something comes from the tissue into the lumen. So when we talk about secretion in terms of like endocrine functioning, we're usually talking about releasing into the bloodstream for a purpose. But when we're talking about the excretory system, secretion is drugs and toxins and things that we don't want that are being put into the lumen to be excreted. Yeah, so the main concept of countercurrent multiplier. So if we follow this descending limb, as we get lower, the outside becomes more solute concentrated. So that water, the water that's in this limb, is going to flow out just by passive transport, because there's high solute in here. It's going to flow out, and that you would expect that to cause, you know, a decrease in the solute concentration. But as it comes over here, we're actually pumping ions back into the medulla. So it's restoring the concentration gradient that the water was pulled out of. So it's going in two different directions, and it's sort of restoring. It's sort of like a cycle. So it has two stops, and they're sort of feeding in and on and off each other. So water goes out on the left side of the diagram, and then as you go up the other one, the ions go out and they sort of restore. The purpose of counter-current multiplier. Yeah, so it's the purpose of the counter-current multiplier is, yeah, it's to keep water flowing back in, and it's also to improve the efficiency. Because if it weren't counter-current, then we would have to use ATP a lot more often for active transport if we weren't sort of setting this up in a way that it would reestablish the gradient. When the body has excess water, we'll talk a little bit about blood volume and excess water, but basically when it has excess water, the distal convoluted tubule is not permeable to water and it just gets excreted with urine. You have very dilute urine and it exits that way. I don't have too many like mnemonics or ways to recall this information. I know a lot of these biology courses, it really is just straight information. So you're kind of just going to have to commit it to memory.